Cytoplasmic determinants include proteins and RNAs, orchestrate an organism's development along with genetic programming. Cell differentiation, cell specialization in structure and function. Morphogenesis, the process by which an organism takes shape and the differentiated cells occupy their appropriate locations. Model organism, a species that lends itself to the study of a particular question, is representative of a larger group, and is easy to grow in the lab. Acrosomal reaction. It begins when a specialized vesicle at the tip of the sperm, called the acrosome, discharges hydrolytic enzymes. Acrosome, a vesicle at the tip of a sperm. Fast block to polyspermy, prevents multiple sperm from fertilizing an egg. Cortical granules, vesicles that fuse with the egg plasma membrane, initiating cortical reaction. Cortical reaction. Cortical granules in the egg fuse with the plasma membrane, the secreted contents clip off sperm-binding receptors and cause the fertilization envelope to form. This acts as a slow block to fertilization envelope, a protective layer that resists entry of additional sperm nuclei. Slow block to polyspermy. The fertilization envelope and other changes in an egg's surface function act as a longer-term barrier to sperm. Zona pellucida. The extracellular matrix of the egg functions as sperm receptor among other things. Cleavage, a succession of rapid cell division. Blastomeres, results from cleavage. The zygote is split into many of these smaller cells, each with its own nucleus. Blastocoel. It is the first 5 to 7 divisions within a fluid-filled cavity during cleavage. Blastula, a hollow ball of cells resulting from the blastocele. Yolk, stored nutrients. Vegetal pole, the concentrated end that yolk gravitates toward. Animal pole, the opposite pole to the vegetal pole. It is also the site where the polar bodies of oogenesis bud from the cell. Gray crescent, a light gray region of cytoplasm located on the side opposite sperm nucleus entry. It serves as a marker for the future dorsal side of the embryo. Meroblastic cleavage. The incomplete division of a yolk-rich egg. Holoblastic cleavage. When a cleavage furrow passes all the way through the cells. Gastrulation. Groups of cells that undergo a morphogenetic process. Gastrulate. The name for the embryo while in gastrulation. Germ layers. The three layers produced by gastrulation are these embryonic tissue layers. Ectoderm forms the outer layer. Endoderm forms the inner layer, lines the embryonic digestive tract. Mesoderm, middle layer, fills the space between the ectoderm and the endoderm. Invagination, the flattening and formation of a vegetal plate that buckles inward as a result of cell shape changes. Archenteron. The open end of this becomes the anus, also called the blastospore. Blastospore will become the anus. Dorsal lip. The part ABOE, the crease, becomes the dorsal side of the blastospore, is called this. Involution. When future endoderm and mesoderm cell layers on the surface of the embryo roll over the edge of the lip into the interior of the embryo. Yolk plug consists of nutrient-laden cells. These protruding cells will move inward as a expansion of the ectoderm causes the blastospore to shrink further. Primitive streak. The pileup of cells moving inward at the blastoderm's middle nib produces this thickening. It runs along the embryo's anterior-posterior axis. It is functionally equivalent to the blastospore in the frog, but the two structures are oriented differently in the two embryos. Organogenesis. When the three embryonic germ layers develop into the rudiments of organs. Notochord. The skeletal rod characteristic of all chordate embryos. Neural tube. Runs along the anterior-posterior axis of the embryo. It will become the animal's central nervous system. Neural crest cells. These cells migrate to various parts of the embryo, forming peripheral nerves, parts of teeth, skull, bones, and so many other different cell types that some developmental biologists have suggested that the neural crest could be considered a fourth germ layer. Somites. These cells are arranged serially on both sides along the length of the notochord. Parts of somites dissociate into mesenchyme cells. Amniotes. The embryos of animals are surrounded by fluid within a case formed by a membrane called the amnion. Reptiles, including birds, and mammals have theirs amnions. Extraembryonic membranes. Membranes located outside the embryo. Coron. Completely surrounds the embryo and the other extraembryonic membranes. Amnion. Eventually encloses the embryo in a protective, fluid-filled amniotic cavity. Yolk sac. Encloses the yolk, which provides nutrients until the time of hatching. Allantois. Disposes of waste products and contributes to gas exchange. Blastocyst. The mammalian version of a blastola. Inner cell mass. A group of cells clustered at one end of the blastocyst cavity. Trophoblast. The outer epithelium of the blastocyst always not contribute to the embryo itself but instead provides support services, such as breaking down molecules of the endometrium. Convergent extension. A type of morphogenetic movement in which the cells of a tissue lower rearrange themselves so that the sheet becomes narrower, converges, while it becomes longer, extends. Cell adhesion molecules. Transmembrane cell surface proteases that bind to CAMs on other cells. They vary in amount, chemical identity, are both from one type of cell to another. These differences help regulate morphogenetic movements and tissue binding. Coherins. An important class of CAMs. It requires calcium ions outside the cell for proper function. They are involved in the tight adhesion of cells in the mammalian embryo that first occurs at the 8-cell stage, where cadherin production begins. Induction. A mechanism where once initial cell asymmetries are set up, subsequent interactions among the embryonic cells influence their fate, usually by causing changes in gene expression. Fate maps. A labor-intensive study to produce useful territorial diagrams of embryonic development. Pattern formation. The development of an animal's spatial organization, the arrangement of organs and tissues in their characteristic places in three-dimensional space. Positional information. The molecular cues that control pattern formation. Apical ectodermal ridge. A lymphoid organizer region that is a thick area of ectoderm at the tip of the bud. Polarizing activity. A second major lymphoid organizer, it blocks mesodermal tissue located underneath the ectoderm, where the posterior side of the bud is attached to the body. Inducers. Certain regions induce changes in adjacent cells and act as organizers or embryonic development. Additional aspects.